Alright, so this video is going to focus on plants that lack the ability to carry out photosynthesis as a result of losing their chloroplasts during the course of evolutionary history. Now what this means is that these plants have actually moved from an autotrophic niche where they fix their own carbon from the atmosphere into sugar using energy from sunlight to a heterotrophic niche where they are reliant on exogenous sugar, both for chemical energy that they harness from the bonds in sugar, as well as carbon for building the backbones of different biological macromolecules that organisms require to carry out the processes of life. Alright, so before I start talking about these ecosystem level interactions between plants and fungi, I thought it would be useful to briefly review the similarities and differences between the light-dependent reactions of photosynthesis and oxygen respiration in heterotrophs. And so this page has a lot of information. It might seem overwhelming, but the important thing is that in both cases, protons are translocated across the cell membrane from the interior of the cell to the exterior of the cell. This translocation generates an electrochemical gradient where high potential energy is outside the cell and low potential energy is inside the cell. Protons are then allowed to translocate back to the interior of the cell along this gradient, which is energetically favorable, and the energy released by this translocation is harnessed by the selective channel through which the protons flow, which is ATP synthase. And so in both cases, life harnesses this energetically favorable reaction, this flow of protons back into the cell, to an energetically unfavorable reaction, ATP synthesis. The important difference between respiration and photosynthesis in this regard is that the electrons that are used in heterotrophic oxygen respiration come from sugar. And so glucose or some other sugar is oxidized to CO2, and that oxidation is coupled to the reduction of NAD plus to NADH. And this NADH is then used as a kind of a reserve of reduction power. In photosynthesis, electrons are instead stripped from water, and then in photosystem 2, it comes first, but it was the second one discovered, I think. In any case, in photosystem 2, the electrons that are stripped from water are energized using energy from the sun, which is transduced by an antenna complex. I'm not going to get into it. But in any case, this electron is given a high potential energy, and it is then dumped onto more and more electronegative shuttles in this membrane. And so the electron is first dumped from photosystem 2 onto this molecule called PQ, or plastoquinone. This plastoquinone, the reduced form of plastoquinone, PQH2, is then oxidized, and that oxidation is coupled to the reduction of this plastocyanin. From here, the electron has dumped its potential energy. The energetically favorable movement of that electron through these different shuttles is used to pump a proton, just like it is in respiration. But in this case, the electron is then re-energized by a different photosystem and dumped onto NADP to form NADPH, which is again it's a way to store reduction potential for use later when it comes to synthesizing sugars or any kind of macromolecule.